Hello. Today's topic is about glycolysis pathway. Let us first understand the term glycolysis. Glyco refers to glucose and lysis means breaking down, so glycolysis means breaking down of glucose molecules. This is a very superficial definition. The detailed definition is breaking down glucose molecule into 2 molecules of pyruvate, 2 molecules of net ATP, 2 NAD molecules that occur in the cytoplasm of the cell. Before we go through the complex pathway, let us first start off with a situation. Imagine you are consuming rice, which is a complex carbohydrate. Rice is digested into the end product, which is glucose. What happens to glucose now? The glucose molecule will then enter the cells. So, the very first thing about glycolysis is the entrance of glucose into the cell. You have remembered that glucose is a relatively large molecule, so it cannot diffuse into the cell. Therefore, it enters the cell via glucose-specific transporters such as GLUT. I will not be going through the different types of glucose transporters in this video because it will be out of topic. However, if you want me to make a video regarding these glucose transporters, comment down or request via the Google form attached in the description below. Now, going back into the topic. The situation now is glucose is already in the cytoplasm of the cell. Next, the enzyme hexokinase will add one phosphate group to carbon number 6 of the glucose. This process of adding one phosphate group is called as phosphorylation. Also, this process of adding one phosphate group requires one molecule of ATP. You also must remember that this step is a rate-limiting step. When phosphate is added by hexokinase, glucose is now transformed into glucose 6-phosphate. Why glucose 6? It is because the phosphate group is added to the 6th carbon. Next, the glucose 6-phosphate rearranges with the help of the enzyme phosphoglycoisomerase into fructose 6-phosphate. Many students struggle to remember the product. The way I would remember is I break down the term. In this case, phospho refers to the phosphate group. Glycoisomerase means isomerize glucose. The isomer of glucose is fructose. By combining these, you can remember the product, which is fructose 6-phosphate. The next step is similar to the first step. So another phosphate group is added to the first carbon of fructose 6-phosphate. This process requires one molecule of ATP and is catalyzed by phosphofructokinase. The product is now called as fructose 1,6-biphosphate. This is another rate-limiting step to be remembered. Now this product needs to be cleaved and the enzyme needed is fructose biphosphate aldolase. An aldolase is a lyase type of enzyme whereby its function is to cleave the substrate. So, the aldolase cleaves fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate has 3 carbon atoms and dihydroxyacetone phosphate also has equal number of carbons. What you need to remember is both glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers. This means they can rearrange to form its counterpart. Keeping this concept in mind, dihydroxyacetone phosphate rearranges or isomerizes with the help of triose-phosphate isomerase to glyceraldehyde 3-phosphate. The steps 1 to 5 referred to energy investment phase because 2 molecules of ATP is used. Remember now you have 2 molecules of glyceraldehyde 3-phosphate. Why? If you know the answer, comment below. Now, using 1 molecule of glyceraldehyde 3-phosphate, we enter the next step. In the next step, 1 inorganic phosphate is attached to carbon 1 of the glyceraldehyde 3-phosphate, converting it into 1,3-bisphosphoglycerate. This reaction also consumes 1 molecule of NADH and is catalyzed by the enzyme glyceraldehyde phosphate dehydrogenase. Next, the enzyme phosphoglycerate kinase will remove 1 phosphate group and attach it into 1 molecule of ADP producing ATP. At the same time, the resultant product is 3-phosphoglycerate. Then the enzyme phosphoglycerate mutase transfers the phosphate group to another carbon atom. What is the meaning? The meaning is since 3-phosphoglycerate has the phosphate group attached to carbon 3, the enzyme detaches the phosphate and reattaches into carbon 2. I remember mutase because this process is like a mutation. Now, the product is 2-phosphoglycerate. The enzyme enolase removes one hydroxyl group from 2-phosphoglycerate, converting it into phosphoenolpyruvate. Lastly, the remaining phosphate group is transferred from phosphoenolpyruvate to one molecule of ADP producing ATP. The phosphoenolpyruvate is also converted to pyruvate. This is the last rate-limiting step. These steps 6 to 10 are known as energy payoff phase. These are the important things that you must know that makes it popular questions in exams. As usual, there are quiz questions. Pause this video and answer them. Comment your answers below. As usual, please like, comment and subscribe if you find this video useful, as this will motivate me into doing more videos like this. Don't forget you can request your biology or medicine topics via the Google form that is linked in the description below. Thank you and see you all in the next video.